Kia ora koutou, my name's John Hosking, I'm the Dean of Science, and it's my great pleasure to welcome you to Waipapa Taumata Rao, University of Auckland, and the inaugural lecture of Professor Tony Hickey. Before I introduce Tony, I'd just like to pause and recognise the passing of Wednesday Davis, one of the SBS technical staff team, who died tragically on Mount Ruapehu last weekend. Our thoughts are with her, whānau, friends and colleagues in this very difficult time. Nā koutou I tangi, nā tātou katoa. When you cry, your tears are shed by us all. We're also here to celebrate Wednesday's life, of course, but also to celebrate the career of Tony Hickey to date. Tony has broad research interests in ecology, physiology, biochemistry and biomedicine. His work is based around the physiology of mitochondria, the energy powerhouses of cells, and particularly what happens to them when they are stressed, which often leads to cell death. Because such processes are so fundamental to life, Tony's work has covered a wide variety of organisms, including fish and marine invertebrates, frogs, birds and insects, and particularly looking at how such organisms adapt to extreme adversity, as well as a broad range of biomedical areas including pancreatitis, shock, and factors affecting heart metabolism. He says the value of this work is that we learn how animals survive and succeed where we cannot, and succeed where we cannot, and it informs us of life's limits and what species will win or lose. Tony did his PhD here in Auckland, after which he became a research scientist, initially with Protemics, and then joined the school as an academic in 2010. He's not only an accomplished researcher, winning a University Early Career Excellence Award, he's also an excellent educator, teaching into the flagship BioSci 101 course with innovative pedagogical approaches that are well received by students. He's a major contributor to the culture of SBS, being awarded a School Citizenship Award in 2021. There's obviously a story there.
More than one.
Tonight, Tony is going to tell us about his journey to become a scientist. He says that if we're lucky, we'll also learn something about mitochondria. Tony, the floor is yours.
All right, kia ora. I'm Tony, as you've just been told, and I sort of struggled with how to attempt to do this, convey what I've done and things. So I popped up the road and I caught up with my mate Bob, and Bob said, what you've got to do is you've got to have a talk outline and you've got to stick to that. And so I said, yeah, Bob, I'll come up with that. And so the first bit will be chaos, the next bit will be chaos, and the last bit will be science chaos, okay? I'm going to put forward something quite new, so it might be kind of a different talk to the normal inaugural lecture. It all really starts with my mum and dad, okay, but let's go back to where they came from. So mum was actually a Scot, born in Edinburgh, and she was born to Etty up here and John over here on the right. Both of them were actually, all the stories I remember of them were during the war. So Etty was a matron in a children's hospital and delivered babies while bombs plummeted down during the Blitz. And John was a doctor, he was a physician in an army hospital in the Sudan and Egypt, where his unit was actually bombed 3 times by Rommel. And we're related to Rob Roy McGregor, so related to a thief. All right, Dad, well, he actually is obviously Irish with the name Hickey, but he was actually born in Wellington, but his parents came from Australia. So we've got Constance and we've got Fergus, and they are descendants of more criminals that were probably exported to Australia, and originated from County Clare, obviously, from the picture there. So anyway, whoops, let's go back. So it was probably 1969 on Waitangi Day. I was actually born in November, so I was mapping back. So I was wondering, Mum, is that correct? Not wanting to put you on the spot. But anyway, there was a twinkle in my dad's eye. And I was born at Tauranga South Hospital, so this is pretty much where I come from. So this is down at Mount Wanganui and things. And here's my family. I'm very lucky, I've got a lovely family. I've got 3 sisters, an older one, Sue, who's not here. She's sunning herself in Bali. And then my twin sisters we've got over here, we've got Sally and we've got Joe. Hopefully I got that right. It was pretty hard to tell them apart, even for me as a kid. Okay, they used to have different freckles and things. All right, so here's me, adorable as ever. And then it all goes downhill. I get uglier and uglier, but you can see that I'm on a bike, okay? Now, Mum and Dad, this is serious, they actually thought I had some sort of mental health problem. They thought I was a bit daft, and so they took me to the psychologist, and this guy gave me like this puzzle, and he said, right, pop outside and you work on this puzzle, I'm going to talk to your parents and go through things. And so I walked outside, and it wasn't actually this, it was actually parts of the Large Hadron Collider. And then a few minutes later I had assembled it and brought it in. And the guy said, whoa, that's so fast. I can't understand how your son did this. Only 2% of the population can put that puzzle together. Your son must be a genius. So I remember the car drive back and I was sitting in the car and they were raving, oh, he's a genius. He said he's a genius. Anyway, Saturday morning they opened up the Herald and there was a story about how their psychologist is actually out mowing his lawns nude. So, yeah. True story. Anyway, Dad was a really good cricketer. He really loved cricket. And he grew up in Taupiri and he was kind of isolated there. And so all his mates got sent off to Wanganui Collegiate. And he always wanted to go to Wanganui Collegiate. But he went off to Hamilton Boys. And so he thought, what would be good for Ant, and this would probably sort him out because he's not doing so well at school, is we'll send him to boarding school. So they sent me to boarding school, and I didn't really appreciate boarding school. I found it quite hard. But one of the things, I excelled in a few subjects, and I say a few, but one of them was biology. And this is a comment from one of my teachers, and I thought it was quite cool actually. I thought it was quite apt, it sort of fitted me. But I did learn not to have outbursts from here on. As you know, I'm sort of the strong, silent type. But while I was at school, I was actually before I went to King's, I kind of fell in love with the sport of cycling. And I started up a cycling team, and I wasn't allowed to actually race my bike originally. I had to actually play hockey or rugby and then go and race. So I had to actually do another sport beforehand. So I'd play hockey and then I'd go off and ride bike races and things. And I was actually really useless at riding a bike. And then when I got to about 16, something happened to me physiologically and I got good at riding a bike. And when I was going 17, there was this guy, Jack Broom, who's this amazing cycle coach. And he got hold of 4 of us guys, and we were like the bad news bears. And he trained us, and somehow we won the national team's pursuit. And this sort of set me off on a path of really getting into cycling. Why do I love the bike? Well, to me, it's got some romance to it. It's a sport where there is a possibility of serious injury, there's suffering, and there's speed, and there's danger. This is one of my great heroes, Bernard Hinault. He won the Tour de France 5 times. His nickname was the Badger because he was so aggressive in racing. And one thing I thought was quite impressive, because I always remember this, he fell off his bike in a sprint finish, and then he got up, and he finished, and then he won the Tour de France with a broken nose and ribs. Really impressive. But one of the best stories to come out of the Tour de France is this one here with Laurent Fignon, who wears glasses and had blonde hair. He inspired me. But the idiot refused to wear an aero helmet and he lost the Tour de France by 8 seconds to Greg LeMond. And that's an amazing story as well. Greg LeMond had been shot the previous year and he had shotgun pellets. He lost his kidney, he lost most of his blood, and he still to this day has pellets in his pericardium. So quite an incredible character. Anyway, so I got into cycling. I was nowhere as good as these guys, but I loved it. And I got to travel all over the place. I made teams and all sorts of things and did all sorts of crazy stuff. The milk race, team time trials, races in New Caledonia, Australia. I went on and on. But one of the cool things is I actually made it into our Olympic team. I didn't actually get to ride the Olympics in Barcelona in 1992, and this sort of broke my heart a wee bit. But, you know, life sucks and then you die. I got to travel all over the place, as I did. We went to the Caribbean, we went throughout Asia, we went all throughout Europe, Australia, Tasmania, sort of Australia. And the place I really loved actually was France. I lived in this place called Troyes, and our team raced all over France. It was really, really good. And I was about 19 or something. And the lifestyle, the countryside, the people, I just really, really loved France. The problem was, when I made the Olympic squad, I couldn't actually stay in France because I had to break sort of contracts and stuff to go and race for New Zealand, so it wasn't going to work. So I went to Belgium, and Belgium was an unusual place. I liked it. It was really exciting, but it tended to look like this a lot when I was there, and that was, it was wet, and there were cobblestones everywhere. Okay, so you tended to fall off a lot, but the cool thing is you could race every single day, and when you raced and you got, say, about in the top 10, you got money, and so I could actually survive by racing. And I tended to, I was very, very skinny if I didn't get anywhere, and so you had to really focus on getting somewhere. It was very, very hard. But we had this crazy guy who looked after us up here in the white shirt with his hand on my shoulder. You can see that I don't really think too much of him. He was, this guy was Staff Boone, and he was like the Tony Soprano of cycling. He was really dodgy, but he got us to race all over the place. And then with the Scotsman on our group here, I could never understand a single word he said. So he took us all over the place again. We raced here, there, everywhere and things. And you know, I had a really, really good time. And there was one thing where I became academic for a little while. For a joke, we dyed our hair black and my IQ doubled. So this is one of the best races I rode. This is the Tour of Tasmania. I actually won this stage. It was the Queen stage, the hardest stage of the tour. I rode away up this big climb. I was unusual. I was quite tall, but I could get up hills. And in the background are Australians. So I've ridden away from Australians. Okay. I had a really good mate actually when I was racing, and here he is, my arm around him. This is Mark Rendell. We were cobbers, so we were the 3 years we flatted in Belgium together. Really got on really, really well, and you know, we'd race for each other. I would lead him out, he would lead me out. We'd chase people down if he was in the yellow jersey or something like that. So we became very, very close friends actually. So you built up a lot of you know, camaraderie. And he actually gave me this. I better pull it out. Or is it So his nickname, so he's Maori, his nickname, he had a pendant, and his nickname became Taniwha. And because my last name's Hickey, they changed it, and my nickname's Honi. And so those were our call signs when we were racing. So he got me a little pendant and had it blessed by his auntie. So there we are. Anyway, about this time, I met the love of my life. Maserati. And my life started to change quite a bit. I started thinking more about, you know, what was going on. And the following year I had a really bad crash. This isn't it, this is just one of many. But I had one where I dislocated a shoulder and it didn't heal very well. And then I had to go and ride some races in Europe and things, and I had a sore throat and it just went downhill and I was losing weight and getting skinny. So I was riding this race in Germany and it was pouring with rain and it was going way too fast, unphysiologically fast I believe. And I just climbed off my bike and got in the wagon and came home. And that's my last race number for New Zealand. So it's kind of weird because I didn't really have that many regrets. I was kind of happy. I was going to change my life and do something different. But I'd given up on cycling, but Cycling New Zealand hadn't given up on me. There was a bit of a problem. Our manager, this guy called John Delore, he was a bit upset because I'd pulled out and the team wasn't doing that well. And so he sort of thought, well, there's a few problems with racing licences. And on our racing licences, we had to list our occupation. And because Kiwi riders usually work part-time jobs, or they have a real job, and French riders, or Germans, or Russians, or Americans, they usually got some bogus occupation. They're in the army or something like that, but they don't usually work. So we would put our own occupations on, and Tim put fighter pilot on his licence, and I had something quite different on my licence. Now my dad was not happy, okay? So what actually happened was that, he was furious actually, because I brought the family name, it was in the Herald, I brought the family name into disrepute, yet I actually was a bit upset with him. He did not value the service industry. And I thought I would have made a great gigolo. Right? Anyway, I had a few other directions. I flattered with this guy who was a cop, and he sort of convinced me, why don't you become a policeman? I said, oh yeah, that's a job. Anyway, I broke my arm before I went down to Poirua. I did all the tests and I was ready to go down, and I broke my arm. So I was running out of options. So I was going down the ranks. I had to think of something good, but it was getting worse and worse. So I thought, how about being a biologist? So I signed up to the University of Auckland, and that was it. I found it incredibly hard, actually. I'd forgotten how to learn. I couldn't write very well. I'm a bit dyslexic, and I'm blonde. So I really struggled. And then after a year, Mark actually won the road race at the Commonwealth Games. And so I was kind of a bit peeved. I was actually working with someone up here, Mike McMurtry, when he did it. We were painting someone's house. I was a little bit peeved, but I was super happy for Mark. But I sort of thought, I could have been there. But, things progress. And I did progress. I made my way through university. I did my bachelor's and then on with my master's. And, I made really, really good friends. And I was inspired by a couple of people. One was this guy here, Associate Professor Dennis Wazell. He was an interesting character. He'd get up on stage and he would tell stories and yarns and things and really get you fascinated. And he taught human physiology, and I topped that class. That was in my second year. And then I came across this guy, Professor Rufus Wells. He's an unusual character, but he told really crazy tales about crocodiles and fish that could breathe through their skin and turtles that could breathe through their arse. It was really quite cool. And then he kind of inspired me to go on. So I did my master's on abalone, which were paua here in New Zealand. And I looked at the muscle structure, if they were growing in rough water or still water or high or low temperatures and things. So I looked at their metabolism. So that was all kind of interesting. But at the end of this, I was sitting in the tea room at SBS, School of Biological Sciences, and I opened up Nature. And in the back of Nature, there was an advertisement to go down to Antarctica. And so I wrote a project up. You had to write a project up and get a letter of reference. So I got a letter of reference from Rufus, and it must have been a cracker because I got to go down. Okay? And I was the only master's student there. Everyone else was either PhD or postdoc or even lecturer level. So I was like the complete newbie. And I had a fantastic time down there. Hopefully this works. We saw all sorts of animals. We got up really close to them. What was really cool was to go out in helicopters, go to the ice edge, and our group would go everywhere and we saw animals, and the other groups went out and they saw nothing. They just saw ice. And so it was really quite cool. I actually played a game of rugby and we won, but I was playing for the Yanks. I didn't say boo for the whole game until the end, and I went and shook hands and said, oh, good on you. And then they realised I was a Kiwi. Anyway, when I was there, I met this guy, Colin Brawner. He's a Canadian, and he was a postdoc then. He's subsequently gone on to take up a position at University of British Columbia. He has a position there that he replaced a guy called Peter Huchachka, who was like basically he wrote the Bible of comparative physiology at the time. So very, very good researcher or scientist. Anyway, I was actually going to do my PhD somewhere else. I was offered when I was down in Antarctica to do one in the States, but it was too hard getting green cards and stuff with my lovely Marie. And so I took up this PhD position with Kendall. And it was on the phylogeography, which is to do with the genetic sort of distribution and geographic distribution all mixed together of these peculiar fish, triple fin fish. So New Zealand is really unique in that we have about 27 to 28 different species of these cool little fish in our waters. And they've evolved, most of them have evolved here in New Zealand. So they're a really cool model to study. Kendall, we got in a car to go up to the Three Kings Islands, and I was with Mike. We just handed in our thesis, and Mike and I went up there, and I jumped on the boat, and Kendall gave me a book, and he said, right, learn all the triple fins before we get to Three Kings. And I learned that triple fins have 3 fins on their back by the time we got there. Yeah. It was pretty good. Anyway, we had some adventures and things, and it was a pretty good time actually, going up to Three Kings and my PhD. We had a really good time doing this sort of stuff. But we also did another trip, Kendall and I. We went to South Africa, and we went to a place called Sodwana Bay, and then we went to Lake Malawi. And we went to Lake Malawi to study these fish called cichlids, which are a bit like triple fins, that they've diverged in these lakes. But it's a real adventure. You know, the locals were fantastic to work with. We actually helped them collect fish. They were like with a conservation group that would collect fish, rear them, and then release them, and also sell some to aquarium things. But Kendall here, you know, it was really quite cool. You know, developed, you know, pretty good relationship. I really realised what a cracker scientist he is. He's really quite remarkable. I also worked with Scott Baker. He was my co-supervisor. And Scott was great. He was great fun. But he tended to have me doing most of the lab work for everyone else because I wasn't a whale person. I was kind of handy. He'd get me to do all the fish, you know, the whale stuff while I was doing my fish stuff. And I also worked with Shane Lavery here. And Shane he taught me so much about statistics and so forth. I was a bit of a numpty here with molecular biology, and I wouldn't have survived without this guy. He was really, really good. Then life events, all right? So after a whirlwind of 7 years, I proposed to Marie on the top of Mount Titirangi, and the ranger came up and said, "You've got to go because we're closing the park." Anyway, it's a bit of a rock and roll sort of situation. And then we went on our honeymoon. We went all over the place. We went to Las Vegas, Grand Canyon, Disneyland, as you do on your honeymoon. And then here we are here. All right, so boom chicka wow wow. Now, I came back to New Zealand, and one of the things I was now on a roll with my PhD, and I was sitting there, and Dick Bellamy rocked on into the computer room where I seem to spend my entire life analysing sequences. And he came in and he said, "Right, you'll do. We need someone to write a bio bulletin. It's like a newspaper thing. We used to have one. It's supposed to be, you know, like a bit of informative, maybe a few jokes, and you can do it." And I said, "Well, do I have a choice?" And he said, "No, you don't." So anyway, I thought, oh, what you got to do is you got to do a really bad job, and they'll never ask you to do it again. Okay? So I thought, yeah, I can do this. Got it sussed. So I came up with my first edition, all right? And there's a bit of a story in here. So the first one is about myself, all right? The second one here is, it's a true story. Dick went to Memphis and there was a bit of soap and there were some curly hairs on the soap, and he took that, and you know how there's DNA sometimes in the follicles of the hair. He took that, he extracted the DNA and he expressed it in cells, and lo and behold, he got Elvis. It's true. But he had a few issues. He was missing a few genes. His hips didn't work properly. But he thought that was a bad thing. I thought this was a new locus. He could make some money out of that. Yep. Anyway, Dick thought this was really good. And he was asking me, he said, "When's the next edition?" And I was like, "Oh, yeah, it's coming." And then he called me one day, and I actually had something rolled up in my hand, which was a draft. And I sort of said, "Well, yeah, I was working on it." And he said, "Well, I'd like to introduce you to the Dean of Science from Cambridge or Oxford or something." I said, "Oh, how do you do, Tony? It's very nice to meet you." And he said, "Have you got the new draft?" I said, "Oh, yeah, I've got it in my hand." And he grabbed it off me and he unrolled it, and it came back with this. Now, there's actually another word under rooster. And I thought if I showed it, I'd get fired. But anyway, I came up with a new gene for this one here. Yeah. Big Dingle 6. All right. Now, that did the trick. I never got asked to do anything again. I tell you what, it was a lot worse when you opened it up and read inside it. No one complained the last time I did it, though. I thought they were thinking they were encouraging me. Anyway, back to New Zealand, triplefin fish. Triplefin fish are really, really amazing. My project, I insisted that I could do some physiology, so I looked at physiological adaptations of these fish, a genome size evolution to see if they'd evolved by a thing called polyploidy, where they've got genome multiplication and things like that, larval dispersal, which was completely boring, and then discovery of some other stuff, which I'll get to now. So one of the big things that came out of my PhD was this paper here. And it was looking at population diversity and the structure around the country. So Kendall and I had gone these road trips, and Marie, and I went to the North Island, Marie and I, and Kendall and I did the South Island, and we collected samples from all over the place, and then we looked at mitochondrial genes. And we looked at how the animals, you know, the different genotypes fell out, haplotypes. And we've got a few interesting things. So one of the main things is we found that as you got deeper in the water, the genetic diversity increased in the fish, and as you got shallower, it got less. But also the population structuring increased as you got shallower. And this we could relate back to glaciation. And particularly the South Island was basically all single haplotypes for some species, which sort of reflected that they'd probably been wiped out and then they'd recolonised from a single sort of fish population. So that was kind of cool. Yawn, yawn, yawn. Now, if we look at one species here, Grahamida capito, I thought this was particularly cool, and I actually won the PhD prize for this because I got some sponsorship from the brewery. But Grahamida capito has now become Forsterigine capito. It had a couple of interesting what we call clades, or little groups. And one of these groups was from Akaroa, and the other group was from the Chatham Islands. And they found out they were very distinct. And I was like, new species. And Kendall was like, nah, I'm a lumper, not a splitter. And I said, but Maui's dolphin has decided on one base pair, and he wouldn't let me still do it. Anyway, I'd lined up sponsorship and everything because my brother-in-law worked for Lion Nathan. And I was going to go with Grahamida spatesii for one of them, and spatesii old darky eye for the other. I figured there could be some money in this for me. But that wasn't the end of it. So one thing is that there was this group where the haplotypes fell out with a totally different species, right? And what this meant is there had been hybridisation. And so there were two species of triple fin that had some hanky-panky, all right? And so this is Varium and this is Capito. And if we think about where they were all found in Fiordland. In Fiordland, you've got this dark layer of water where it's harder to see, and it's also colder because it's ice melt, and so you get deep water species coming up. And so these two species had kind of intermixed, and we had hybrids. Now, to me, this was fascinating because it was probably the first example of bear goggles in fish, all right? And I came up with a gene with that as well, actually, so Fugly fix it too. Okay, so anyway, this is the end of my PhD, and I moved on. And I was contracted to do some work on trying to keep fish alive, this crazy thing with these chambers where we pump them up with the air or different gases, and they seem to survive quite a long time. I had a few explosions. One put a massive dent in Mum's Saab. I didn't actually realise, it almost killed me actually, this big shard of plastic in the garage wall, about heart height. But that dent in the car actually got blamed on my brother-in-law, who's sitting here now, actually. But I took this idea to Anthony Phillips, and I said, hey, this could be a useful thing for transplant, a way of transporting organs. We could keep fish alive for this amount of time. Maybe we can push gas into these organs and keep them going. Anyway, it was a total fizz of the grant we wrote together. But Ant said, you know what, we need someone like you working at Proteimics. So I don't know, you think I was going to blow something up. Anyway, he put me onto Garth. And Garth, we used to call behind his back G-Force. And he was probably the best boss you could ever have, okay? And I had this job interview with him. I came in and I sort of at the end of it, I said, why on earth do you want me? You know, I've worked on fish and stuff like that. And he said, well, it doesn't really matter. They all sort of work the same. But one of the things, it's not what you know, it's how you think. Okay, now Garth is a remarkable man. He's actually my kāmatua. I'll go to him for advice on stuff, personal stuff, all sorts of things. He's a fantastic man and a bit of a brain as well. But I did wonder why he picked me, because the next day I think we were at a Christmas party and this was how I was dressed as a bandit. Okay, so life changes. This time here, sprogs. Okay, so we had kids and I can remember working for Garth. He got me doing stuff on mitochondria, which was just the best opportunity for me. And I was just absolutely, had the tremors, and I thought I've poisoned myself, because with mitochondria you work with all sorts of things like cyanide and rotenone and oligomycin and things. I thought, oh, I've poisoned myself. And I went to the doctor and I was shaking, and the doctor said, oh, you had any life changes? I said, oh, we just had a baby boy. He said, go home and get some sleep. The tremors went, remarkably, they went. They came back though. Anyway, some fantastic times, and we've taken our boys all over the world with friends and things, and we've taken them to Austria for work I do with a group over there. And now they've grown up into fine young men. And one of the things though is when you travel with them, you often get in predicaments that I find are You know, it can be a little bit dodgy. You know, say you're swimming somewhere and you could get in trouble. There's always a good way out, I've found, and that is put on a foreign accent, it's particularly the German accent. Ich möchte schwimmen auf dem Norden. And anyway, speaking of German accents, Jörg decided to take me on as a lecturer. And you know, there's this position there, and it was a bit of a sitter. Actually, Kerry was in my interview panel, and was sitting there, and Jörg asked this question: where do you see yourself in 5 years' time, Tony? And I said, your seat, mate. I don't want it, I tell you. It's a hard job. Now, I talk about teaching. Someone very special here, Mandy sitting down here, she's been incredible. So I was put in to teach 101, and 101 is a massive paper, thousands of emails per week, and it's just mind-bogglingly difficult to do. So I'm co-director, and I would say basically I don't do much co. It's mostly Mandy. The teaching's actually quite intense. I do love it. It's a fantastic sort of environment to do that. Now, my other love is this paper, BioSci 335, which is comparative physiology. And here we do all sorts of stuff. We look at the locomotion of how fish move, and I even invented a robotic fish that generates force as it swims, and you can swap out the tails and see what happens when you change the tails and change the speed and forces and stuff. And all sorts of other things. But tragically, the person helping me out with a lot of this was our dear Wednesday. And so we've lost her. But one of the things that I will always remember her by, she actually sent me the fish thing. I had to find it the other day. It was quite hard. But one of the things was we were rearing anemones for a lab, and she kept sending me updates on how many babies they had. And she said that I had had 172 babies. I should be a proud father. They had my eyes. But looking at them, they must be bloodshot. Okay. So to you, Wednesday, with all our love. So, return to Tiddlers of Mystery. At this point in time, using some equipment that Garth had paid for, we didn't know about this, I did some work with Zoe Hilton. I ended up co-supervising her with Kendall, and we looked at some physiological effects of triple fins. And we found out that their cardiac mitochondria were very robust to temperature if they lived in a rock pool versus if they were a deep water species. And this was actually kind of cool. And what was really cool about it is I used it to get a Marsden fast start. And that led me a lot further along. One of the things it did is it got me to think up some new gear. So these machines had these windows which are like eyes. And I figured that I could actually do something with this, where I made some sensors that you could plug into the front. And I made them out of bits of duct tape, hose clips, LEDs, photodiodes, which are like little solar panels, and just batteries and stuff. And I managed to make these super sensitive sensors that could measure fluorescent things, such as free radicals, fluorophores that work with ATP, pH, all sorts of crazy things. Okay. Now, another company got hold of this, and it's been a very productive thing for me because he's paid for me to go over and work in workshops with him, take our family, all sorts of things. And this is Eric Niger, he lives in Innsbruck in Austria, and we got to go skiing and stuff in the Alps. And it was really good. So I thought I could market my stuff pretty much the same, and my marketing team needs to be fired. Anyway, I used this to look at shark metabolism, and one of the things here is this shark here is peculiar. It's up in the Philippines, it's called a bamboo shark. In Australia, it's called the epaulet shark, and it's called the epaulet shark because you can see the spots on it. But the cool thing about this shark is that, well, A, it can kind of walk on its fins like a lizard, and it will do this when the water's out to get back into the water. But it stays on the reef when the tide goes out, and often it can get stranded, and it's without oxygen for a bit. So it can go for 4 hours at 30 degrees in anoxia, and anoxia is no oxygen. It gets no brain damage, and it has no problems with its cardiac function. It just shuts down and turns back on. So this is amazing to study for things like reperfusion injury and surgery and things like this, and ischemia where there's lack of blood supply. And so I got encouraged to go over and work with Colin Brawner from down in Antarctica. So he asked me to come over, and so I came over, and I met this other guy who made a comment about, what does it take to be a good scientist? And I said, a crazy shirt. And I'm just looking at his shirt there. Yeah, that's pretty crazy. But I also met this lovely woman, Gillian Renshaw, who's sitting down here at the front. And we've done all sorts of stuff since then. Lovely woman. And I'm just going to destroy that by saying there's someone even better. And that's David Attenborough. She worked with David Attenborough. And there's actually, it's on YouTube where she's talking about the sharks and stuff. And so she tells these amazing stories about this fantastic man who I think has done the most probably for communicating climate change and things out of anyone. He's just such a good man. Anyway, I use these sensors to do other stuff. I looked at diabetic stuff, and this is looking at what happens to the efficiency of diabetic hearts. And surprise, surprise, it does. The efficiency drops off, it gets really bad, and it's quite miserable if you're a diabetic heart or a diabetic rat. We also did other things that weren't related to directly to mitochondria to start with, but we worked out how bumblebees actually warm up. We almost got this into nature. They sat on it for a month before saying, oh no, you could try our sister journal. And we worked out how they make heat, and we made our own system too. We made a calorimeter out of bits of TVs, old chiller boxes, stuff we stole from other people's labs. I've got a habit of doing that. And it worked. We couldn't believe it worked. Anyway, we worked that out. About this time too, this French guy called Jules Devers emailed me, actually emailed Neil Herbert, and he forwarded on to me. And Jules wanted to do his Masters, and he was funded by Rotary. So he came out to New Zealand, did his Masters with me, and then he went back. And the data he got from his study, again, on the wonderful triple fins, was enough to get me another Marsden grant. And so, really, really cool stuff. Now, Jules is really quite handy, actually. Oh, the other thing, too, is this work actually got us this grant. It was with Nigel as well, who sadly, we've lost Nigel a while back. But Jules is very handy. He made this system here, and this was actually to work on another Marsden I got later on, and this is to measure, we could measure heart function in a perfused heart, and this is a fish heart beating away, and we could shine lights through this heart of different sort of intensities or wavelengths or colours, and then we had sensors where we could pick up the light coming through it, being transmitted through it, and we could work out what was going on with the mitochondria inside the heart as we heated it up until it failed. And some really, really cool data. And the cool thing is, you might have noticed there was lots of Lego in there. So we use Lego for a very serious reason, and that it's actually metric, and it's very well made and machined or moulded, and you can pull it all apart, put it into a little box, get to Heron Island or wherever you're going, put it all back together, and you've got a system. And it's a lot cheaper than a lot of the other systems you'd get, say, in bioengineering. Science time. Now, Bob told me, you've got to present some proper science, Tony, otherwise people might doubt that you're any good. So I'm going to do some science. And this is a little bit different to what you would normally expect. So this is my mate Bob here. I'm going to talk about hot mitochondria and a vital force. So, as animals get too hot, what happens is that they tend to die. Why do they die? It's usually heart failure, usually have a heart attack, and this includes us. When there are, high temperatures, people often have strokes and stuff, and heart attacks usually are part of that. So why the heart? Well, I reckon actually the brain might fail as well, but the heart has to pump continuously, obviously, or you're dead, and in a human heart it pumps about 7 tonnes a day if we think of the mass of water, so that's an awful lot. Hearts tend to be very sensitive to temperature, and it appears that something is going on in terms of ATP supply. And 90% of ATP comes from what gives you power, kids? Definitely not love. Mitochondria give you power, okay? And here's a pretty picture of a mitochondria. Most cells in animals have a mitochondria, or nearly all cells. Red blood cells in us don't, okay? So ATP, I talk about ATP, some people here won't know what ATP is. So ATP is adenosine triphosphate. That made it easy, didn't it? Anyway, so what it is this is a molecule of ATP, and on here we've got 3 phosphates, so the triphosphate means 3 phosphates. And these are actually charged. And the last phosphate can be kind of blown off the end. And when this happens, it releases quite a lot of energy. And that energy can be used to drive muscle contraction, the myosin heads and muscles, to drive ion channels, all sorts of things. Things that do work. Now, you can push it back on as well, but that takes the same amount of energy that it released to put it back on. And that is kind of a complicated process. So where do we get the energy to do that? Well, we take food, we digest it, we extract the energy, and what I mean by extracting is we pull electrons out of that food, and we use those electrons to drive proton pumps, okay? And those electrons then flow off to oxygen, which then go to form water, so it's a nice, safe sort of waste product. But the protons go off to drive turbines, real turbines, okay? And those turbines are found in the membranes of the mitochondria. And one thing that's kind of really important for here, these are the membranes that have the turbines in them and the proton pumps, all right? And they're the same width here as they are here. And this is a heart mitochondria and this is a liver mitochondria. What is a proton? It's this big red thing. It's positively charged, unlike an electron, which is negatively charged. And electrons are used to drive pumps here. They pump the protons out. And the paradigm is that it builds up a gradient on the outside of a bacterium or the outside of the membrane of a mitochondria. And then they make their way back through this ATP synthase, which is a turbine. And this was the concept and the kind of discovery of Peter Mitchell, who was awarded a Nobel Prize in 1978 for this. So effectively what's happening is the turbine is turned by protons binding to little subunits within this thing called the C ring, that orange bit at the bottom. And they go around and they make it turn, and then they're ejected on the other side. And at the top, what's happening is that an ADP and a phosphate go in and they get mooshed together, and that's the force that pushes them back together to make them ATP, and then it can go off and do stuff. And I see the protons as being like a bicycle chain, simply because bicycles are really cool. Okay, back on topic. So what does this have to do with hot fish hearts? So this is kind of a complicated plot. It's what we call an Arrhenius plot. It's where you can try and determine the break point in, say, a function of an enzyme or something. You can use it for looking at heart function as well. And it take me ages to explain it, but basically, trust me, the heart of this fish fails, which is a spotted wrasse, a common thing you'd get off a pier here. It fails at about 27.5 degrees, right? So we know that it fails there. Now, this is work done by Fatima Iftikhar that was a fantastic student of mine, my first PhD student, and she then did some mitochondrial work. Now, how do we measure mitochondria? Well, remember that oxygen is used in respiration, okay, so we need oxygen. And that use of oxygen is kind of proportional to the amount of ATP that is produced. So if we measure the oxygen going through the mitochondria or being used by tissue, we can infer what's going on in terms of ATP synthesis, you would think. Okay? So what we can do is we can take tissue from an animal, take it out, and we can process it in different ways, and we can bung it into these machines called oxygraphs, and we can measure the rate of oxygen uptake. And we can plot that here. The blue line here is the oxygen concentration dropping as the tissue respires. And the red line is us looking at the conversion of that blue line into a rate, what we call a flux, or the rate of oxygen being consumed. And so we can see it here changing as we add all sorts of different fuels in. These different fuels represent what's going on inside your cells. Okay? Now we've got lots of different machines to do this. They're all heroes apart from SpongeBob. Jules painted these faces on these machines. I don't know what he was thinking about with SpongeBob. Okay. Now, if we look at respiration and temperature, we can see here that as temperature increases, it's what you'd predict. The rate increases in terms of the oxygen uptake. And this is very common for cold-bodied animals like fish or lizards and things. But one of the things we can see here, there's kind of a plateau here, and that was statistically significant, but it's all a bit cagey. So what we did is we used that fluorescence system to measure ATP synthesis, and we got a totally different effect. What we got is that it didn't actually increase as temperature went up, which is kind of odd. We didn't do the same temperatures, though, the lower temperatures. But then when we got to about 25 degrees, it suddenly started to drop. Okay, and this is fundamentally important to an animal because you need ATP. Now, if you think about what's actually going on, the respiration rate is going up, you're using lots of oxygen, but the ATP synthesis rate is crashing. And this means that you're using lots of oxygen to make hardly any ATP. So your efficiency is going through the floor. And this is probably what is stopping the heart. It runs out of ATP. Your heart only has 60 seconds of ATP before it stops if you don't have oxygen. Okay? All right. So what actually makes the fish, we know the heart fails because of the mitochondria. What makes the mitochondria fail? And this was what the title was of my latest Marsden. So the dogma is that the inner mitochondrial membrane has issues and it becomes leaky to these protons at elevated temperatures. But is this actually true? Let's first look again at how mitochondria work. So here's a mitochondria with lots and lots of membranes and cristae and so forth. If we zoom in on them, we can see these little dots, and these dots are all in a line. Now, they've used some sophisticated methods like cryo-electron microscopy to actually zoom in and look at what's there. And they've then reconstituted the images they've got, and they've worked out that you've got these nodules here, and these are the ATP synthases. Now, ATP synthases, from a potato to a human, all arrange in this V-like structure, and they form folds on the mitochondrial membrane. They form the ridges on cristae, okay, which are the membranes inside the mitochondria. And this is another vision of it, where we've got them all lined up in a row. And the purple and blue objects are actually what we call respiratory complexes, and we'll just call them pumps for now, okay? So they are proton pumps, and they will pump protons through the membrane, and then they make their way through the ATP synthases. Now, why are they arranged like this? So this is actually missing a set of proteins down the end here that actually closes the cristae off. So we'll call this loop of membrane the cristae. Karen Davies, an amazing researcher from the States, she actually came up with this concept along with some Germans, and we've got a proton pump here, and we can see that the protons are being pumped from the matrix here into what we call the intermembrane space, the IMS. So it goes through here, and then somehow miraculously it makes its way to the ATP synthase and pops out. Now, the typical paradigm here is it's to do with a pH gradient. Okay, so there's a high concentration of protons in that gap, and that concentration is driving the protons through the ATP synthase. And so this is another way of looking at it, where I've made a cheesy cartoon, and your protons are making their way through the ATP synthases. And they're going at enormous rates, really, really, really fast. Okay? Now, if you heat a mitochondria up, the paradigm is the membranes all of a sudden become leaky. The lipid molecules start to move really, really fast, and protons can sneak through. In fact, they think that water can actually make its way through the membrane. Water shouldn't go through membranes, but they think it can make what they call water wires, and that conducts the protons through. So the protons go through, and they just get pumped and pumped again, but they don't go through the ATP synthase. The ATP synthase doesn't turn, and you die. Okay? Now, Peter Mitchell contended that what was actually happening was that protons were being pumped into the inner membrane space, and they were building up, and they were generating what we call a low pH or microdomain, or a voltage difference across this. And in fact, it's about 0.14 of a volt across the membrane, and we can actually measure that. But if you actually work out the pH, calculate from the pH, and you do the math, you can actually count how many protons there are in an entire mitochondria, and it's about 8. Okay? Sounds crazy, sounds like a trick of numbers. But if you actually work back the number of protons that are in a litre of water at pH 7 and so forth, it's ridiculously low. And then you think of the volume of a mitochondria, it's 1 femtoliter. Okay? But we know, I've actually calculated this, known respiration rates and how many protons are flowing. 1 million protons are flowing in a single mitochondria every second. So where are they in this mitochondria? Well, it brings us to this. So some physicists, some real scientists, actually did some work where they were looking at interfaces of membranes and what was going on. And if we look at what's actually zoom in on here, and we look at it, in the intermembrane space, we have water, okay? We also have proteins and things here as well. And this is a dangerous place to be if you're an unlucky little proton, because you'll get snaffled. And also it's a three-dimensional space, and you'll go off all over the place. But if we get closer to a membrane, what happens is that the water molecules start to align, and they actually form an ordered state, and it is called the Stern layer, and it has a nice German name, and because it is German, it probably has some rules. So one of the things is that this Stern layer lines up, and the protons can actually skip from water molecule to water molecule in one direction. There's a really cool reason for this as well. But it makes its way from the pump to the ATP synthase, and it does this really, really rapidly. But what's important is the distance from here, from the pump to the ATP synthase, has to be quite small. All right? So there's a membrane model of how protons actually move. And the stern layer has rules. It orientates the water. The bulk water phase is in 3 dimensions and it's chaotic. So I told you there would be some chaos. The stern layer diffusion is 2-dimensional. So all of a sudden the protons are restricted to they've lost that other dimension. The distance between the pump and the ATP synthase is actually quite small. So that means that they're being attracted towards the ATP synthase. Well, that's what this group contends. And with this attractor, diffusion is now limited to 1 dimension. Okay? So what we've got here is we've got a very fast transport system next to the membrane. And also this layer here is sensitive to temperature, which is kind of interesting. So let's look at what happens with a fish. This is a banded wrasse, very much like the spotted wrasse, quite common around New Zealand. But we're looking at mitochondria now being incubated at different temperatures, and it's getting hotter and hotter and hotter. And what happens when they get hotter and hotter and hotter is the membranes start to lose their structure and they start to bulge. So that got us thinking, what's happening to these stern layers and how the protons are moving. The other thing we can do is you can just take a green LED and shine it through mitochondria and heat them up. And the light scatters and you can measure the scattering of the light. Very cheap and effective way of looking at mitochondrial structure. And here we're looking at a temperate banded wrasse and a tropical wrasse, which we got from Heron Island. We did some work up there, and we can see that the mitochondria are actually swelling as temperature rises. And there's a difference between a tropical and a temperate fish, which is comforting. All right. So what's actually happening here? So this is our standard model of how the protons are flowing. Now what happens is it gets bigger. Okay, we swell those mitochondria like we saw. Well, one of the things is that the distance between the pump and the ATP synthase is going to increase. And this means that the proton is hanging around on the membrane and may not actually get to the ATP synthase. So that's one big problem. And we can see here in this little plot, I did some mathematics, and you can actually work out how long it takes for a proton to move around on the membrane. The next thing too is that this thing called the dielectric constant, which is how permissive the environment is for the proton to move through it. It actually changes with temperature. Okay, so as it gets hotter, the dielectric constant of the bulk phase, the water, drops. So that means that it's easier for the proton to go from the membrane into the water. Okay, and then it's lost forever. So to sum it up, what we've actually got here is we've got a nice closed system here. Everything's working. It's all hunky-dory. It's awesome. And then we heat it up and it all turns to custard. And the distance gets greater. The chances of forming water wires increase, and so maybe things are leaking through from one side to the other. And then the other is that maybe the protons are now being lost up into the watery layer. So we could actually test this, and all we had to do was get mitochondria, heat them up, and put a pH indicator into the media, and we could measure changes in pH. So we did exactly that. And what we see is we heat up, heat up, heat up, nothing really much happens, and then all of a sudden, above 23 degrees, it starts to drop. And so this is evidence that the stern layer is probably breaking down. It's actually quite a simple experiment, which is kind of cool. So we just got this just the other day. All right. And I would say in German, that is wunderbar. Okay. So where now? This has actually changed how I think things actually happen. So I'm going to go out on a bit of a limb here, and this is kind of odd. So what actually propels protons, right? Are they pushed or are they pulled? One of the things too is it actually electrostatic forces? It's something that hasn't been considered that much in terms of how protons move around. And this is my little boy Fred, and you can see here that static electricity actually has profound effects. And we're looking at this at a macro scale, not at a molecular scale. You can see here it's being pushed apart just due to electrical charges. So can these effects also explain how the mitochondria works in its structure? All right. So I'm proposing something quite new. So if we put like charges together, they repel, don't they? Think about what's happening at one of these proton pumps where the protons are being pumped out crazy. They're really high concentrations. One of the things is that you can put some mass to this, you can use Coulomb's law, and you can actually estimate what's probably going on. So I was looking at the total amount of protons coming out per second, and then how they would actually dilute as they went through the waters around this super complex or proton pump. And you can see that the actual force between the protons, because you can calculate the force between them, actually drops off as you get further and further away. That makes total sense, doesn't it? Now, that got us to thinking about, or I sat down, I thought, well, I could come up with a model to try and explain this. So I sat down with Tinkercad and I drew something up and I came up with a different way of thinking of this. Now, think about if we've got one proton pump and it starts pumping. The protons are coming out and they're pushing off each other, right? But if you've got one pump there, you don't really get that much power and they're not quite making it to the ATP synthases, right? Now, we put two there. You've got one proton pump pushing off the other, and you've got lots and lots of protons, you've got a higher concentration. The other thing too is because you've got two there, you get an additive effect, so you get amplification, all right? Now, I've calculated how many of these pumps you have on the flat part of the mitochondria, and it's around about 20, right? So when you put a lot there, you really start driving the protons, and they can really go quite fast. And you get a summative effect of the forces, so you get this massive amount of force. And this is all electrostatic forces. It's not to do with pH. Okay? Now, if we get an expanding membrane, we're stretching the membrane apart and the supercomplexes or the pumps are being pulled further and further apart, so we also lessen that effect. And you can do this just by summing up the number of charges per water molecule. And if you actually put the forces into here, which is kind of a bit dodgy to do, you start to get massive effects because it's non-linear. You start to get a huge effect to do with if the membranes are stretching in the mitochondria. All right. Now, one last thing before I finish up here is that, remember I said that the membranes, let's say a liver mitochondria doesn't actually have to work that hard. It's there processing lots of things that are going on inside your cell. It's not necessarily just making ATP. But a cardiac mitochondria is basically there just to make tons of ATP for you. And so a cardiac mitochondria has a lot of cristae and the liver mitochondria has fewer. But if you look at the cristae, they're about the same dimensions in terms of how thick they are. And this got me wondering about something kind of cool. And I think this is kind of neat. Now, if we zoom in on this tomograph, this tomograph we've got of a mitochondria, which is three-dimensional electron microscopy image, and we look there, that's actually looking into a cristae, and that's the inner membrane space in that hole. Okay? What we're actually doing is we're bringing around two membranes like this, and we've got pump here, and they're actually pumping protons into each other. So we're actually magnifying the amount of protons in between them, and that gives us a lot of driving force, right? And so there we are there. Now, what if we swell them? We stop all of that, and we lose that power, okay? And you can actually, again, you can calculate what the forces will be from the known amount of protons on these respiratory complexes. So, can electrostatic forces explain mitochondrial structure? Well, I think they can, but hypotheses need to be tested to death. And this is one of the beauties of science, is that we have to put questions out there, not be afraid to put them out, and test them to bits where you can be wrong. The person that came up with this was Sir Peter Mitchell, the one who came up with the concept of oxidative phosphorylation. A fantastic character, actually, a very good story if you want to look him up in terms of the struggle in science. Anyway, that's kind of it. But I've got to thank some people for where I've been. Luckily, Alice wasn't dribbling in this picture. I was a bit annoyed, actually. I was hoping she'd have a big bit of goob. But this is from Heron Island. We had a great time over there. We've got Jules' special research bit of equipment was a paddleboard. Yeah, it wasn't a nudist beach, actually. And lots of people I need to thank. I've probably missed some people, so apologies for that. Some special people, Professor John Windsor's here. He gave me lots of breaks, actually. Got into HRCs and stuff like that that helped me out with career advancement and things. And David Crossman, basically for being a good mate. He's been with me all the way through from being a master's student. Fantastic guy. Jiwon Hong, we'd be lost without Jiwon. Absolutely wonderful person. And she looks after us, actually, and tells me off most of the time. And Ant, who's been best of friends and just a great scientist to work with. So, I've got a bit of French. Je me sens à l'intérieur, et le truc comme ça, which translates to feel inside and stuff like that. I got that from Flight of the Conchords. Feelings. Ugh. Okay. Anyway, the one person I really need to thank is my lovely wife. We've got the most magnificent family. Very lucky. Things have gone well for us. Our boys are beautiful, both young and now, and so's my wife. I love you.